In this video, we will step through the process of translation and highlight the key steps. Translation is composed of three steps shown here. Recall that these are the same names for the steps in transcription. So lucky for you, there's no need to memorize new step names, just new processes. The steps of translation occur out in the cytoplasm, involve our three types of RNA, tRNA, rRNA, and of course, mRNA, and translate that mRNA into an amino acid sequence that is then modified into a working protein by subsequent steps. Initiation is the first step in translation, and per usual, we know this process best in bacteria. In bacteria, the ribosomal RNA recognizes a conserved sequence called the Shine-Dalgarno sequence, which is the ribosomal binding site. This binding is via hydrogen bonds and complementary base pairing. Notice this sequence is just upstream from our start codon. At the outset of translation, our ribosome is not yet assembled and only assembles after several steps of initiation. Once the Shine-Dalgarno sequence and 16S-R RNA bind to one another, three different initiation factors are involved in binding the initiator tRNA, which recognizes the start codon. This initiator tRNA carries a modified methionine amino acid. Once this initiator tRNA is attached, the large ribosomal subunit binds and translation can begin. In eukaryotes, this process is similar, with the same start codon, but in eukaryotes, the ribosomes recognize the 5' cap that we put on the mRNA after transcription concluded. A few key differences in eukaryote initiation include different initiation factor complexes that target the 5' cap. These bind to the small subunit of the ribosome and begin scanning for the start codon. Once that start codon is found, the large subunit joins, and we have an assembled ribosome that can begin translation. Once translation is initiated, we can go through the processive steps of building an amino acid chain from the red codons in the mRNA. The basic process illustrated here involves recognition and initial capture of tRNAs at the A site of the ribosome. This then slides into the P site, where the newly arrived amino acid is added to the growing chain of amino acids. Once this amino acid is added to that growing chain, the now uncharged tRNA slides into the E site, where it is subsequently ejected. tRNAs proceed through this process, depositing their amino acids in an ever-lengthening chain in the P site. This process continues until we reach one of three stop codons. These codons do not code for amino acids, but rather release factors, which have a similar shape to tRNAs, but do not carry amino acids. The binding of a release factor stops translation and dissociates this whole complex. Let's close this video with a couple points on bacteria that show just how amazing they are. Given that bacteria lack a nucleus, transcription and translation can happen simultaneously. Once enough mRNA is created in transcription, ribosomes can bind to it and begin translation. This is known as coupling. Eukaryotes can't do this because transcription and translation are segregated into the nucleus and cytoplasm, respectively. However, since eukaryotes do a lot of post-transcriptional modification of those mRNAs, this segregation is actually a good thing.